Dear viewers, this video is part 3 in the 4-part series on cancer cell biology in the Medicine for Anyone in YouTube channel. Role of P53. If DNA damage is detected, the P53 pathway is normally activated. Called the guardian of the genome, P53 is a transcription factor that is normally present in the cell in very low levels. This level is generally regulated through its rapid turnover. Normally, P53 is bound to MDM2, a ubiquitin ligase that both inhibits P53 transcriptional activation and also targets P53 for degradation in the proteasome. When DNA damage is sensed, the ATM, that is ataxia-telangiectasia mutated pathway, is activated. ATM phosphorylates MDM2, releasing it from its inhibitory bond to P53. P53 then stops cell cycle progression, directs the synthesis of repair enzymes, or if the damage is too great, initiates apoptosis, that is programmed cell death of the cell to prevent the propagation of a damaged cell. Inducers of P53 include hypoxemia, DNA damage caused by ultraviolet radiation, gamma radiation, or chemotherapy, ribonucleotide depletion, and telomere shortening. A second mechanism of P53 induction is activated by oncogenes such as MYC, which promotes aberrant G1S transition. This oncogene checkpoint leads to the death or senescence, an irreversible arrest in G1 of the cell cycle of renegade cells that attempt to enter S phase without appropriate physiologic signals. Senescent cells have been identified in patients whose pre-malignant lesions harbor activated oncogenes, for instance, dysplastic nevi that encode an activated form of BRAF, demonstrating that induction of senescence is a protective mechanism that operates in humans to prevent the outgrowth of neoplastic cells. Not surprisingly, given its critical role in controlling cell cycle progression, mutations in the gene for P53 on chromosome 17p are among the most frequent mutations in human cancers. Most commonly, these mutations are acquired in the malignant tissue in one allele, and the second allele is inactivated, leaving the cell unprotected from DNA damaging agents or activated oncogenes. Some environmental exposures produce signature mutations in P53. For example, aflatoxin exposure leads to mutation of arginine to serine at codon 249 and leads to hepatocellular carcinoma. In rare instances, P53 mutations are in the germline, as in Lee-Fraumeni syndrome, and produce a familial cancer syndrome. Another mechanism for inactivation of P53 in malignant cells is due to alterations in regulators such as overexpression of the inhibitory MDM2 protein. Whether inactivated by mutation or inhibited by regulatory factors, absence of normal P53 function leads to chromosomal instability and accumulation of DNA damage, including acquisition of properties that give the abnormal cell a proliferative and survival advantage. Like RB dysfunction, most cancers have mechanisms that disable the P53 pathway. Indeed, the importance of P53 and RB in the development of cancer is underscored by the neoplastic transformation mechanism of human papillomavirus. This virus has two main oncogenes, E6 and E7. E6 acts to increase the rapid turnover of P53, and E7 acts to inhibit RB function. Inhibition of these two targets is required for transformation of epithelial cells. Another cell cycle checkpoint exists when the cell is undergoing division, that is M phase. This is the spindle checkpoint, which acts to ensure that there is proper attachment of chromosomes to the mitotic spindle before progression through the cell cycle can occur. If the spindle apparatus does not properly align the chromosomes for division, if the chromosome number is abnormal, or if the centromeres are not properly paired with their duplicated partners, then the cell initiates a cell death pathway to prevent the production of aneuploid progeny, that is, having an altered number of chromosomes. Abnormalities in the spindle checkpoint facilitate the development of aneuploidy, which is frequently found in cancers. In some tumors, aneuploidy is a predominant genetic feature. So, dear viewers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. In other tumors, a defect in the cell's ability to repair errors in the DNA, such as due to mutation in genes coding for the proteins critical for mismatched DNA repair, is the primary genetic lesion. Cancer cells can have defects in any of several DNA repair pathways in addition to mismatch repair, including deficient interstrand crosslink, double strand breaks, single strand breaks, base excision, nucleotide excision, and translesional synthesis. Mismatch repair. This describes cells that have mutations, that is, changes in certain genes that are involved in correcting mistakes made when DNA is copied in a cell. Mismatch repair MMR deficient cells usually have many DNA mutations which may lead to cancer. Interstrand crosslinks. Normally, the two nucleotides on opposite DNA strands are covalently joined by weak hydrogen bonds. DNA interstrand crosslinks, ICLs, are lesions caused by a variety of endogenous metabolites, environmental exposures, and cancer chemotherapeutic agents that have two reactive groups. The common feature of these diverse lesions is that two nucleotides on opposite strands are covalently joined. Interstrand crosslinks, ICLs, are highly toxic DNA lesions that prevent transcription and replication by inhibiting DNA strand separation. Agents that induce ICLs were one of the earliest and are still the most widely used forms of chemotherapeutic drug. Single strand break. DNA, that is double-stranded DNA, is a double-stranded helical structure. Any damage or break at one of these strands is termed a single strand break. Here, the nucleotide sequence of only one strand is altered. It breaks the whole sugar phosphate backbone of the DNA. This type of DNA damage is the most common one. Factors such as UV rays, radiations, chemicals, etc. can cause this damage. Double strand break. It is the less common type of DNA break seen in organisms. Here, both the strands of the DNA are altered or broken. This also results in the breaking of the sugar phosphate backbone. It is difficult to repair this damage by normal cellular mechanisms. Thus, they can also be lethal. The broken strands can lead to mutations. Translesion synthesis, TLS. Translesion synthesis, TLS, is a pathway in which specific polymerases, that is TLS polymerase, are recruited to the replication machinery and perform synthesis across the DNA damage. In general, tumors have either defects in chromosome number or defective DNA repair pathways, but not both. Defects that lead to cancer include abnormal cell cycle checkpoints, inadequate DNA repair, and failure to preserve genome integrity, leading to DNA damage. These defects and the stress of the resultant increased DNA damage make cancer cells more vulnerable to additional DNA damage, which can be exploited by chemotherapy, radiation therapy, targeted therapy, and immunotherapy, the major systemic therapeutic approaches effective against cancer. Alternatively, research is ongoing in an attempt to therapeutically restore the defects in cell cycle regulation and DNA repair that characterize cancer, although this remains a challenging problem because it is much more difficult to restore normal biologic function than to inhibit abnormal function of proteins driving cell proliferation, such as occurs with oncogenes. Cellular senescence, the irreversible cessation of growth of normal cells while the cells remain viable, has been termed cellular senescence. When normal cells are artificially grown in laboratory, most cells are not capable of sustained growth. They quickly reach a point where they either undergo cell death due to excessive DNA damage or other factors, or they become senescent. Fibroblasts are an exception to this rule. When they are cultured, fibroblasts may divide 30 to 50 times and then they undergo what has been termed a crisis, during which the majority of cells stop dividing and enter senescence. This form of senescence is termed replicative senescence. Many other cells die and a small fraction of emerge that have acquired genetic and epigenetic changes that permit their uncontrolled growth. Among the cellular changes during in vitro propagation is telomere shortening. DNA polymerase is unable to replicate the tips of chromosomes, resulting in the loss of DNA at the specialized ends of chromosomes, called telomeres, with each replication cycle. At birth, human telomeres are 15 to 20 kb pairs long, kilobytes. They are associated with specialized telomere binding proteins to form a T-loop structure that protects the ends of chromosomes from being mistakenly recognized as damaged. The loss of telomeric repeats with each cell division cycle causes gradual telomere shortening, leading to growth arrest when one or more critically short telomeres trigger a p53 regulated DNA damage checkpoint response. Cell death usually ensues when the unprotected ends of chromosomes that lead to chromosome fusions or other catastrophic DNA rearrangements. Cells with certain abnormalities, such as those with non-functional PRB and p53, can bypass this growth arrest. The ability to bypass telomere-based growth limitations is thought to be a critical step in the evolution of most malignancies. This occurs by reactivation of telomerase expression in cancer cells. Telomerase is an enzyme that adds TTAGG repeats onto the ends of chromosomes. Most normal somatic cells do not express sufficient telomerase to prevent telomere attrition with each cell division. Exceptions include stem cells, such as those found in hematopoietic tissues, gut and skin epithelium, and germ cells, that require extensive cell division to maintain tissue homeostasis. More than 90% of human cancers express high levels of telomerase that prevent telomere shortening to critical levels and allow indefinite cell proliferation. Major efforts are underway to develop methods to inhibit telomerase activity in cancer cells. For example, the protein component of telomerase, HTERT, may act as one of the most widely expressed tumor-associated antigens and can be targeted by vaccine approaches. However, a caveat to targeting telomerase for anti-cancer treatment is the potential for inhibiting its activity in certain normal cells, such as stem cells, required for maintaining the normal physiologic state. Although most of the functions of telomerase relate to cell division, it also has other, several other effects, including interfering with the differentiated functions of at least certain stem cells. Much remains to be learned about how telomere shortening and telomere maintenance are related to human illness in general and cancer in particular. A variety of other stresses on cells, both environmental and intrinsic, including radiation, chemotherapy, reactive oxygen species, and oncogenic mutations, can also lead to senescence, primarily those that induce DNA damage similar to that seen in cells with shortened telomeres. Signal transduction pathways in cancer cells. Signals that affect cell behavior come from 1. Adjacent cells, the stroma in which the cells are located, hormonal signals that originate remotely, and the cells themselves, that is, autocrine signaling. These signals generally exert their influence on the receiving cell through activation of signal transduction pathways that have as their end result the induction of activated transcription factors that mediate a change in cell behavior or function, or the acquisition of effector machinery to accomplish a new task. Although signal transduction pathways can lead to a wide variety of outcomes, many such pathways rely on cascades of signals that sequentially activate different proteins or glycoproteins and lipids or glycolipids, and the activation steps often involve the addition or removal of one or more phosphate groups on a downstream target. Other chemical changes can result from signal transduction pathways, but reversible phosphorylation and dephosphorylation play a major role. Proteins that add phosphate groups to other molecules, proteins, lipids, or nucleic acids, are called kinases. Two major classes of kinases involved in signal transduction pathways important for cancer cells are tyrosine kinases that phosphorylate tyrosine and serine threonine kinases that phosphorylate serine threonine, either directly or indirectly. However, some kinases can phosphorylate both, such as the MEK kinases that can phosphorylate both tyrosine and threonine. Phosphatases, that is protein tyrosine phosphatases and protein serine threonine phosphatases, remove the phosphate groups to reverse the kinase activity. Various kinases play critical roles in signal transduction pathways important for malignant cells. These include a number of receptor tyrosine kinases, RTKs, as well as various protein kinases, both tyrosine and serine threonine kinases, downstream of receptors that transmit the signals within the cell. Although pathways are depicted as distinct, there are complex interactions between pathways within cells. Tyrosine is converted by phosphorylation by tyrosine kinase or MEK kinases, and the enzyme reaction is inhibited by protein tyrosine phosphatases. Serine threonine is phosphorylated by serine threonine kinases or MEK kinases, but inhibited by protein serine threonine phosphatases. Normally, kinase activity is short-lived and reversed by protein phosphatases. However, in many human cancers, RTKs or components of their downstream pathways are activated by mutation, gene amplification, or chromosomal translocations to have enhanced and or prolonged activity. Because these pathways are important in regulating proliferation, survival, migration, and angiogenesis, they have been identified as important targets for cancer therapeutics. Inhibition of kinase activity is effective in the treatment of a number of neoplasms. So, dear viewers, this is the end of part 3 in the 4-part series on cancer cell biology in the Medicine for Anyone in YouTube channel. Thank you for watching the video.